We will solve another 2023 exam, a recent one, June. So we did the Novembers. Now let's start. What are two characteristics of all living organisms? Breathing and reproduction, photosynthesis and excretion, reproduction and respiration, or respiration and photosynthesis?
C.
Yes.
Reproduction and
Okay. If you
respiration.
noticed, always the first question is about characteristics of living organisms, and the second question is always about classification. Here, the photograph shows an organism. It's obviously a bird. Which visible feature can be used to classify that this is a bird? This is so easy.
Feathers.
A, of course, birds. These are the only organisms actually that have feathers. If you think of any other organism, they have fur, not feathers. Okay, which statement about cell structure is correct? An animal cell has a cell wall, a bacterium has a cell wall, an animal cell has chloroplast, or a bacterium has chloroplast. Again, this is so clear.
B.
B.
Yes, a bacterium has a cell wall. 4. The diameter of an egg cell from a frog is 2.5. The diameter of what? Of an egg cell from a frog is 2.5 millimeters. The diameter of an egg cell from a human is 120 micrometers. Which value shows approximately how many times larger an egg cell from a frog is than an egg cell from a human? First, are the units the same?
No.
No. So we have to make the units the same, either both millimeters or both micrometers. But if we have a smaller unit, we always make both units the smaller unit. So how do we convert the 2.5 to micrometers?
Divide by 1,000.
Multiply
Times.
by 1. Yes, multiply by 1,000. So 2.5 times 1,000 is 2,500 micrometers. So how many times larger the egg cell is from the frog egg?
We're going to divide.
21.
Yes, how did you get 21?
I divide 2,500 by 120.
Yes, okay. It's going to be 20.8 or times 21. So 2,500 divided by 120. This is going to be 20.8, or it's going to be 21 times bigger. So the answer is times 21. So 4B. 5. When a plant cell is put in pure water, it, 1, water by osmosis and becomes 2. Which words should be used to fill gaps 1 and 2 to compensate or to complete the sentence?
B.
Yes, it will gain water by osmosis and become turgid. Very good. So it's B. We want to get the first 20, the first 30 all correct without mistakes. The last 10 are the confusing ones. The graph shows the results of an investigation into the effect of oxygen on the uptake of glucose by cells. So on the x-axis, you have the time and the mass of the glucose taken into the cells on the y-axis. And the first graph here when there is no oxygen, and the second graph when there is oxygen. Which conclusion can be made about these data? Now, glucose only enters the cells by active transport, or glucose only enters the cells by diffusion, glucose enters the cells by both active transport and diffusion, and glucose enters the cells by osmosis.
C.
C.
Okay, it is C. Now, why did you choose C? Now, let's look at D. Glucose enters the cell by osmosis. Is this correct?
No.
No, definitely not. C is the most logic answer. Okay, nothing enters the cell by only one method. And especially if it's by active transport, it has to start by diffusion and then it follows by active transport. But look at the graph. Here, with time, the glucose does enter the cell when there is no oxygen. So here it shows that the glucose will enter the cell when no oxygen is needed. So the glucose will enter the cell by diffusion. But when there is oxygen, more glucose will enter the cell. So this shows that the glucose will enter the cell when there is no oxygen, and also it will enter the cell when there is oxygen, but more will enter the cell. So this shows that the glucose will enter the cell both by diffusion and active transport. So the answer is C. Do you understand number 6?
Yes.
Yes.
Okay. 7. The table shows the results of food tests carried out on a fruit. The test either Benedict or burette or ethanol or iodine, and the result positive for Benedict and burette, negative for ethanol, and negative for iodine. So what did the fruit contain? This is so easy.
Protein and reducing sugar.
Yes, okay. Benedict is positive, so reducing sugar. Burette is positive, so protein. What describes the structure of DNA? Each strand contains chemicals called fatty acids. Each strand contains a sequence of amino acids. Each strand contains messenger RNA, or two strands coil to form a double helix. So obvious.
D.
What is 8? Yes, 8D. Okay. 9. The statements are about enzymes. 1. Enzymes are catalysts. Enzymes are proteins. Enzymes are used up during the chemical reaction. Which statements are correct? Now, this is so easy.
B.
B.
Yes. Okay. The last one, or number 3, is wrong. So it's 9B. 10. When the temperature increases, the rate of an enzyme catalyzed reaction increases until the optimum temperature is reached. Which statement explains why the rate increases? Nice question. A. The enzyme and substrate have more kinetic energy and collide less frequency, or the enzyme and substrate have less kinetic energy and collide more frequently, or the enzyme and substrate have more kinetic energy and collide more frequently. What's the answer? And the enzyme and substrate have less kinetic energy and collide
C.
less frequently. Yes, obviously it's C. Okay, easy exam. The plant with variated leaves. Now, what are variated leaves? We took it in plants.
The leaves
Covering
have
the
different
plant through
colors.
distortion.
Yes. Variated leaves are leaves that have two colors, white and green. The green has chlorophyll, and the lighter one, or the lighter part of the leaf, which is D, for example, it has no chlorophyll. They were placed in a dark cupboard for 48 hours so that all the starch was removed from the leaves. So this is de-starching. They de-starched the plant. The variated leaves have green parts and white parts. So they're nice, and they told you. Black paper was then fixed on one leaf and the plant was exposed to the light. After 24 hours, which region of the leaf contained starch? Now, this is also easy. Which part of the leaf will contain starch? A, B, C, or D?
C.
Yes, the green part that was not covered. If anyone has any problems, stop me. The graph shows the rate of photosynthesis with increasing light intensity. So the light intensity on the X and the rate of photosynthesis on the Y. Where is light intensity a limiting factor for photosynthesis?
D.
D? Between 3 and 4? How come? Between 3 and 4, the light
A.
intensity is increasing, so it's not a limiting factor. Even though the light intensity is increasing, the rate of photosynthesis is constant. So it is not D. It is in this part of the graph, between 1 and 2. So it's A. When the graph increases and then it becomes constant. This part of the graph, what is the limiting factor? Whatever is on the x-axis, because I'm increasing it. And as I am increasing it, the rate of photosynthesis is increasing. So this is telling me that it was the limiting factor. So this part of the graph, between 1 and 2, okay, the light intensity is the limiting factor. Is it clear now for everyone? Or anyone is confused?
No, it's clear.
Okay, let me just. Good. In humans, which two components of the diet can be broken down to release energy? Carbohydrate and fat, carbohydrate and mineral ions, protein and water, vitamins and protein.
A.
Yes. Okay. Water does not give you energy, and minerals and ions, they do not give you energy. They don't have an energy value. Which is a part of the small intestine? Colon, ileum, esophagus, and rectum.
B.
B.
Yes, of course. Easy. We want to score the highest grade in paper 2. Paper 2 is very easy. It just needs practice. Again, I'm telling you, don't study anything. Don't go back to your notes, except if something is really difficult or you don't understand yet. You just solve as much as possible. 15. Which combination of environmental factors will cause the largest increase in the transpiration rate in a plant? Wind speed, humidity, temperature, light intensity. Choose A, B, C, or D.
B.
Yes, it is B. Why? Because all of them should be high except the humidity, which should be low. The wind speed is high, temperature high, light intensity is high, and the humidity is low. Okay, 16. The diagrams represent the movement of substances between plant cells. The arrows indicate the direction the substance is moving in. Which diagram of translocation is correctly labeled? Can someone remind me what translocation is?
Is the transport of sucrose, glucose and sucrose to different parts of the plant?
Glucose and sucrose? No, there's something wrong
Sucrose
in
and
your
amino
answer.
acids.
Yes, it is the sucrose and the amino acids that are moving in different parts of the plant. They are moving from where to where?
Sources to sinks.
From the source to the sink. Very good. So I want you to look closely at diagrams A, B, C, and D and tell me what is the correct diagram. Which is the correct diagram showing you the movement of sucrose from the source to the sink?
C.
Okay, and back? Yes, it is C. Now, do you all understand why it is C? Let's look at C. Here you have an arrow, this arrow. It's moving from the source to the sink. And sucrose is the correct assimilate here. It's moving from the source to the sink. This diagram correctly shows what translocation is. A is wrong. Why A is wrong? Let me change color. A is wrong because the diagram is correct or the arrow is correct. It is showing you that it is moving from the source to the sink, but it is not glucose. It is sucrose. B is also wrong because the arrow is incorrect. The arrow is showing you that the movement is taking place from the sink to the source. So this is wrong. D is wrong. Why is D wrong? Because again, the arrow is showing you that the movement is from the sink to the source, which is wrong. So the only correct answer is C. Are you all with me? This is a nice question. Clear?
Yes.
Okay, 17. Yes, my dear. You have a question?
No, I was just saying. No.
Okay, good. 17. Which label identifies the right ventricle?
D.
Yes, it's D. This is the right atrium. And this is the right ventricle. And this is the left side of the heart. Good. Okay. The diagrams show sections through three types of blood vessel. They are not drawn to the same scale, 1 to 3. In which order will red blood cells flow through these vessels in the human body when traveling from the aorta to the right atrium of the heart? Now, this can be tricky. The questions will start being tricky.
B.
What did you say?
B.
Yes, correct. Now, why did you choose B? Tell me.
Because the blood is going to first flow through the arteries, which have a narrow lumen,
Perfect. And then?
and then they're going to branch into arterioles, and then going to become capillaries and turn into capillaries.
Excellent. Okay. And then the last
The
thing, they
exchange
will
of
be connected into?
the veins.
Yes. Perfect. Now, if you remember the very popular picture of the capillary bed, you have the artery. It will branch into smaller arterioles that will branch into capillaries, okay? And they will provide the body cells with nutrients and oxygen and glucose, etc., and take the waste products, the urea and the carbon dioxide, and the excess waste products, and they will collect them into veins so that they go back to the heart. So you have artery, arteriole, just like your friend said, very good. Artery, it will branch into a smaller arteriole, and then capillaries, and then venules, and then veins. Perfect. So 18 is B. I'm going to highlight this question so that you go back and look at it. Now, 19. When a person is given a vaccine, a harmless pathogen is injected. The pathogen has which causes lymphocytes to produce. This is an example of immunity. Which words should be used to fill gaps 1, 2, 3 to complete the sentences? What do you think?
A.
Okay, someone said A. Let me see if A is correct. Now, the pathogen has antigens, correct? Okay, which cause lymphocytes to produce antibodies. Of course, lymphocytes produce antibodies. And this is an example of active immunity. Yes. Okay, now the questions, they start to become tricky. 20. What happens when a person breathes in? The external intercostal muscles and the internal intercostal muscles contract. The external intercostal and the internal intercostal both relax. The external intercostal relax and the internal intercostal muscles contract, or the external intercostal and the internal intercostal relax. What's 20?
D.
Excuse me?
D.
Yes, it is D. So here we are breathing in. So this is inhalation. So the external intercostal and the diaphragm, if you remember, they will both contract. The diaphragm will become flat in shape so that we increase the volume and reduce the pressure, and we push or pull air in. And the external intercostal muscles, they will contract so that they push the rib cage up and out so that the ribs, in case they ask you, move up and out. Again, for the same reason, so that the volume inside your chest increases and the pressure decreases, and you breathe or you take air in. Now, as the external intercostal muscles contract, the internal will relax because they are antagonistic. And we always study it this way, that the diaphragm and the external intercostal muscles, they're friends. They either contract together or they relax together. So good, we revised the muscles in your respiratory system. 21. Which graph shows the effect of temperature on enzyme activity? This
C.
graph is very popular.
C.
A, B, C, or D?
C.
Yes.
C.
Yes, we're very familiar with this graph. After running a fast race, some students have pain in their leg muscles due to the buildup of lactic acid. A student wrote this explanation. During the race, the cells did not have enough oxygen for aerobic respiration. 2, anaerobic respiration occurs, so some energy is released from glucose. 3, the lactic acid is produced by aerobic respiration. Which statements explain why the lactic acid builds up in the muscles?
One only.
And what about 2? Anaerobic respiration occurs. Yes. So some energy is released from glucose. 2 is right. 1 is right and 2 is right. 1, during the race, the cells did not have enough oxygen. Correct, of course. 2, anaerobic respiration, because we don't have oxygen. So we will do anaerobic respiration and we'll break down glucose into lactic acid, which is the incomplete breakdown of glucose. And then this lactic acid, after the exercise is over, is going to be oxidized in the liver. It will continue breaking down in the liver into carbon dioxide and water. Remember? Hello? So whoever
Yes.
said 1 only, now you know why it is A, it is not B.
Yes.
Okay, good. 23. What is the name of the process that produces urea and the organ where urea is formed? So the process producing urea, what is its name? And it happens in which organ?
Deamination, and it happens in the liver. C.
Yes. Okay, deamination, and it happens in the liver, correct. 24. The diagram shows the gap between two neurons. What is X? Easy.
Neurotransmitter.
Yes, A. It's neurotransmitter. Which statements are correct for cone cells in the eye? Cone cells are located in the fovea. Cone cells detect color. Cone cells are more sensitive to light than rods.
A.
No, it's B. Okay? Because they did not say which light. They're trying to confuse you. They said 3. 1 and 2, they are obviously correct. Cone cells are located in the fovea. Yes. Cone cells detect color. Yes. 3. Cone cells are more sensitive to light. Which light? Dim light or bright light? They did not say. Because rods are also sensitive to light. But which type of light? Dim light. So these are both photosensitive cells. They are both sensitive to light, but one is sensitive to bright light and one is sensitive to dim light. So 1 and 2 are correct. 3 is not correct. So the answer is B, 25B. Let's go check. 1 minute. This is the mic scheme. This is June 23, paper 23, correct. 25B. Yes. Didn't we say B? 25B. So here they're trying to confuse you. They are very tricky. I'm going to also highlight this question. Okay. So that you go back and look. Okay. 26. Which structure constricts to reduce heat loss? We want to reduce heat loss.
A.
A is a capillary. Capillaries, do they have muscles in their walls? Do they constrict and they dilate?
No.
No. We want something to prevent the entrance of the blood into A. Because if the blood goes to A, it will be very close to the surface of the skin and heat will be lost by radiation. We don't want the blood to go to A, or we want to reduce the blood flow to A. So what will constrict to reduce blood flow to A?
C.
C?
Shunt vessel. B.
B. We want B to close. Why? Imagine the blood is coming in this direction. When B is closed, blood will not enter and reach the capillaries. So less heat will be lost by radiation. Okay? It cannot be D because D is a sweat gland. It's not even a blood vessel. It cannot be A because A is a capillary, and we know that capillaries, they don't have muscles in their walls. They are one cell thick, so they cannot dilate or constrict. We have C and we have B. C is incorrect because C is the blood supply to the hair shaft. It's got nothing to do with heat loss, okay? So the C is a blood vessel, yes, but this is the blood that is supplying nutrients to the hair shaft. It's got nothing to do with loss of heat by radiation. The only answer is B. We want to constrict B so that we would reduce the blood flow entering through B, and in turn reduce the blood flow entering into A, or the capillaries, and reducing radiation of heat and reducing heat loss. Is 26 clear? 26 is B. 26 is B. Okay, now you understand why? I will also mark
Yes.
26, or by default, the last 10 questions are always the tricky ones, or maybe a little bit more than 10. Where are the hormones estrogen and progesterone produced during pregnancy? Amniotic sac, oviducts, placenta, umbilical cord.
C.
Placenta.
Yes, placenta. Okay. Mainly it's the progesterone, but some estrogen is always produced actually the whole time. 28. The umbilical artery takes blood from the fetus to the placenta. The umbilical vein carries blood from the placenta to the fetus. How do the contents of the umbilical artery differ from those of the umbilical vein? Again, the umbilical artery takes blood from the fetus to the placenta, and the umbilical vein carries blood from the placenta to the fetus. How do the contents of the umbilical artery differ from those of the umbilical vein?
D.
Is it D?
Yes. The carbon dioxide, more carbon dioxide. Again, look at the question. How do the contents of the artery differ from those of the umbilical vein? Now they want to confuse you also, because here they said artery and vein. So you will put in mind that the artery will have more oxygen and less CO2, and the vein will have more CO2. This is true. This is in your body. But the situation here is different. You have the artery taking blood from the fetus to the placenta. What does the fetus give the mother? The fetus gives the mother its waste products. So the artery here will have more carbon dioxide and more urea, more waste products. And the umbilical vein carries blood from the placenta to the fetus. The placenta is the mother. So what does the mother give to its fetus? More oxygen and more nutrients. So the answer, yes, it is D. Why? Because the artery will have more carbon dioxide, less oxygen, and more urea. The artery will have more waste products. Is number 28 clear?
Yes.
29. Which row shows some of the features involved in natural selection? Look at it and give me an answer. Natural selection. Many offspring are produced. Humans choose. Yes, humans, they don't choose, correct? Alleles are passed on to the offspring? Yes. Okay, so it's D. Why it is not A? Because alleles are passed on to offspring. No, it's wrong. No, alleles are passed. So A is wrong. The answer is D. Okay, and it cannot be B or C because the humans here choose desirable features. There is a check mark. No, it's not the humans. It's natural. 30. Which substance is coded for by a length of DNA? Now, this is so easy.
Base. A.
Of course, not A. The DNA
D.
is codes for proteins. And which one of those is a protein? The enzyme, the lipase. It has bases, yes, but these bases, they code for proteins. Do you understand why you understand 30 now?
No, not yet.
Okay, the DNA is made up of nucleotides. These nucleotides, they have bases, yes? And we said every 3 bases code for 1 amino acid. Don't you
Yes.
know that? And a
Yes.
length of DNA codes for a protein. So, which substance is coded for by a length of DNA? A protein. Which one of those is a protein? A or B or C or D? A base is not a protein. A base is part of the DNA, yes, but it's not a protein. Glucose is not a protein. Glycerol is not a protein. Glycerol is a carbohydrate. Lipase is an enzyme, and all enzymes are proteins. So the answer is D. Did you get this now?
Yes.
Okay, good.
Thank you.
31. The diagram shows the sex chromosome of a woman and of a man. Their genotypes for a recessive sex-linked condition are also shown. So this is a female because XX, this is XX, and she is a carrier. And this is a male. How did I know? Because I see one long chromosome, which is X, and one short chromosome, which is Y. So this is a male, and he is sick. He has the recessive allele. So you have a carrier female crossed with a male with one recessive allele. What are the chances that their daughter will show the sex-linked condition? I cannot answer this question except if I do a Punnett square. So I am going to do a Punnett square. I'm going to erase this part, okay? And I'm going to use my pencil and draw here a small Punnett square so that I can answer this question. Here I have a mother that is a carrier. She is A capital, A small, and a father, X, Y, that has one recessive allele here. Okay. Now this will be a carrier female, and this will be a sick female because she will have homozygous recessive alleles. And this will be a male that is healthy. And this will be a male that is what? Sick. But they're asking for what are the chances that their daughter will show the sex-linked condition? So what are the chances?
C.
25.
We said C or B?
I guess B. عشان B هي الشغل هي اللي بتشغل
They have 2 daughters and 2 boys. Okay? They have, so they have 2 daughters and 2 boys. One out of the 2 will be sick. So they have two girls and 2 boys. Now they're asking about the girls. Leave the boys now. One out of the 2 girls will be sick. So it is not B. It is C. 31 is C. Let's look at the mark scheme. 31 C. Did you understand why? Because they are playing around with the words. Their question was, what are the chances that their daughter will show the sex-linked condition? If they wrote the question in this format, what are their chances? What are the chances that one of their children will show the sex-linked condition? There will be a different answer. Or what are the chances that one of their daughters will show the sex-linked condition? Because they said the word daughter. So how many daughters do you have in total? You have 2 daughters. And only one out of those 2 will show the disease. So this is 50%. So the answer is C. Did you understand the question? Who is
Yes.
confused? If they had asked you the question in a different format, if they had asked you, for example, what are the chances that one of their children will show the sex-linked condition? Now we have here 4 children, and 2 of them show sickness, which is 1 girl and 1 boy. This is 2 out of 4. So the answer will always be 50%, actually, no matter which format they asked you. If they said, what are the chances that their daughter will show the sex-linked condition? You will check how many daughters. I have 2 daughters, and 1 out of the 2 daughters will be sick. So it is a 50% chance. If they asked you, what are their chances that their son will show the sex-linked condition? I have 2 boys, and 1 boy becomes sick. So it is also 1 out of 2. So the answer is always 50%. Did you understand why? Is 31 clear? Hello? Did I lose you? Can you all hear me? Hello?
Yes.
Yes.
Did you understand this question? Okay, answer back. Is 31 clear for everyone?
Yes.
Okay, now let's go to 32. Which Punnett square shows a test cross that identifies a heterozygous parent? A, B, C, or D? A test cross and the parent is heterozygous. Is it A? Is A correct?
Yes.
No, of course, A is not correct. What is a test cross? A test cross means I have an unknown parent that will be crossed with homozygous recessive. And they're telling me that this unknown parent is heterozygous. So this unknown parent has to be T capital T small. A is 2 or are 2 heterozygous parents. So A is wrong. Are you
C.
with me?
C. Yes.
Of course it's C. Why? Because I have 1 heterozygous parent and 1 homozygous recessive parent, exactly a test cross. Clear?
طب هو
We
أنا
don't
إزاي
want
عرفت
mistakes.
البيرنت
Mm.
التاني homozygous ده
أهو.
recessive.
Because it's a test cross. Now this is telling me that you don't know what a test cross is. What is a test cross? How many times did we study a test cross? A test cross is a cross in order to test the genotype of an unknown parent. You don't know what's a test cross? What's a test cross? A test cross. A test cross is a cross in which we will combine or allow the breeding of a homozygous recessive parent with an unknown parent. We don't know the genotype of this unknown parent. Why do we do that? Because we want to know the genotype of this unknown parent. They told you that this unknown parent is heterozygous. So the Punnett square should be between a heterozygous parent and a homozygous recessive parent. Now you're asking me, how did I know that this other parent is homozygous recessive? Because it's a test cross. It's obvious that you don't know what a test cross is, that you answered and stepped in paper 4 without knowing what a test cross is. Why do we do a test cross? We take an unknown parent of unknown genotype and we cross it with a homozygous recessive parent and we look at the phenotype of the children. If all the children show the dominant allele, then this unknown parent was homozygous dominant. If some children show the dominant and some children show the recessive, then this unknown parent was heterozygous. You don't know what's a test cross? Hello? Clear now?
Yes.
Okay, 33. What is the definition of variation? Now, this is very easy. Check it out. What is variation? Having 2 identical alleles or individuals of the same species in one area, or differences between individuals of the same species, or the way in which new alleles are formed.
C.
Of course, it's C. Yeah, 34. What is a feature of some xerophytes? Large air spaces in the tissues, leaves are rolled up and covered with hairs, or leaves with stomata on the upper surface, or thin cuticle.
B.
Of course, it's B. Okay. 35. Where does the energy being transferred along a food chain originally come from?
Producer.
What's the main source of energy?
The sun.
Yes, of course. 5 more. Which diagram shows a pyramid of biomass for a forest? And don't tell me B. B is not a pyramid of biomass. B is a pyramid of number. The inverted one is the pyramid of number. So it's A, B, C, or D?
A.
A. Pyramid of biomass. It's white at the base and it narrows down just like a pyramid of energy. The difference is that the pyramid of energy looks like a triangle. They draw it like this, and the pyramid of biomass are separate bars. And we even learned how to draw it. 37, we're very close. Which substance is an enzyme used in fruit juice production?
C.
Now, either you know it or you don't.
C.
Yes, to break down the pectin and make juice extraction easier. The diagram shows some of the effects of human activity on a river. So I have fish dying here from lack of oxygen. I have aquatic plants. They grow very quickly, of course, because there must be excess of nitrates. Bacteria use up dissolved oxygen. There has to be a lot of decomposition going on. What could have caused these effects? Air pollution, deforestation, overuse of fertilizers, or presence of herbicides. Now, this is too easy.
Overuse of fertilizer.
Overuse of fertilizers.
Of course. Yes. 2 more. Genetic modification has been used to produce human insulin in bacterial cells. What is inserted into the bacterial cell to produce human insulin? A human nucleus, human plasmids, recombinant insulin, or recombinant plasmids?
D.
Yes, recombinant plasmid. We'll take the gene, we will take the plasmid, we'll fuse them together. It will be part human, part bacteria. This is the recombinant plasmid, and we put it in a new bacteria and then in a fermenter. The last question. Penicillin is made in a fermenter by growing organisms. Which type of organism is used in the production of penicillin? Bacterium, fungus, protoctist, or virus?
Bacterium.
Bacteria.
Fungus.
Protoctus.
Fungus. This is one of the functions of fungi. We use it to make medicine. And the first medicine that was made in history was the penicillin antibiotic made from the penicillium. By the way, the name penicillium and fungus and how it's done, we took it. It's in your notes. We put this penicillium fungus in the fermenter, we make it happy, and once the food resources start to decrease, it starts to produce the penicillin antibiotic. Remember, or you forgot everything?
No, I remember.
Okay, good. We finished this exam. By the way, I hope you get an exam like this. This is very easy. The number of questions that were tricky were not a lot. Please go back and revise those that have been highlighted. When you come to solve paper 2, when you get a correct answer, finish. You got it correct. The wrong answers, go back and understand them. And I am very happy with what you're doing. Yes, you are bombarding me with questions, but anytime you don't understand the question, take a picture and send it to me. Clear? We solve another one or on Tuesday?
Tuesday.
Okay. We solved one together, solve another one today. This will be at least 2 per day, and 2 tomorrow, and 2 on Tuesday. See you Tuesday. Bye-bye.
Bye, Miss.
Bye.